Hello, good morning, everyone. Today I'm going to talk about um, the isomerism in monosaccharides. Compounds that have the same molecular formula but exist in different forms, we call them iso- isomers. They cannot superimpose the molecules. So, structural isomerism, compounds that has the same molecular formula but exist in different structures. Glucose and fructose, they have the same molecular but they have different structures. Glucose is aldehyde while fructose is a uh, ketone. So glucose and fructose are structural isomers. Stereoisomerism or optical isomerism, when a carbon atom is bonded to four different groups, then that carbon atom is said to be an asymmetric center or a chiral carbon atom. So we know that a symmetric carbon or a chiral carbon means that it has four different groups attached to it. Molecules that carry asymmetric carbon centers or chiral carbon atoms shows optical isomerism. So what is optical isomerism or stereoisomerism? It means they cause plane polarized light to rotate in left or right direction. If this when this plane polarized light is is directed towards its molecule or the compound, it will rotate. The light will rotate. So if the light can either rotate in left or right. So when if an isomer rotates the light in left direction, we call it L isomer. If the molecule rotates the plane polarized light into the right direction, we call it a D isomer. L isomer is the mirror image of D isomer. So L and D are mirror images. But we know that mirror images are not superimposed, cannot be superimposed, right? So L and D molecules also cannot be superimposed. For example, this is the L1, this is the L amino acid, this is the G amino acid. So here the H is on this side, NH3 is on this side. Now H is on this side and NH3 on this side, but they cannot be superimposed. So DL, designation of optically active monosaccharides, orientation of O hydroxyl and H groups around the atom adjacent to the terminal primary alcohol carbon in the Fischer projection. Orientation of hydroxyl and hydrogen groups around the carbon atom adjacent to the terminal primary alcohol carbon in the Fischer projection. When the OH group is on the right, it's a D isomer. When it's on the left, it's an L isomer. So what here this slide mean is that we got this chiral carbon. So chiral carbon here is this carbon is a chiral carbon here. So the hydroxyl group attached to the chiral carbon, if it is on the left side, so this is the right side. If it's on the right side, we call it the T glucose. When this chiral carbon this hydroxyl group attached to the chiral carbon is on the left side, we call it the L glucose. So always this chiral carbon is the adjacent carbon to the terminal primary alcohol carbon. So this is terminal primary alcohol carbon. It's on the terminal end. It's also a primary, making a primary alcohol, right? Because on one hydroxyl group is, so we call it terminal primary alcohol carbon. So chiral carbon is always the adjacent carbon to this terminal primary alcohol carbon. Why the G isomer? All the G and L isomers have analytical energies, they have analytical energies. The former monosaccharides predominate in nature. So what's the reason for this is that it's a preference is established, that preference that is established based upon an initial random choice in early evolution. Preference is established based upon an initial random choice in early evolution. So it's something that's been decided like uh, within the initial random choice. It's a random choice that is established in the early evolution. So it was happening like from the beginning of the earth. So now let's go to cyclic monosaccharides. So monosaccharides of 5, 6, 7 carbons. They are more stable in aqueous solution as cyclic structures than they are as open chains. So cyclic monosaccharides form spontaneously due to the intramolecular reactions occurring between the carbonyl carbon and the hydroxyl group attached to the same carbon backbone. So cyclic monosaccharides are formed. That happen kind of spontaneously. The reason is that we don't have to supply energy; it happens spontaneously. The reason is that due to the intramolecular interactions occurring between the carbonyl carbon and the hydroxyl group attached to the same carbon backbone, and the hydroxyl group attached to the same carbon backbone. So carbonyl carbon and the hydroxyl group attached to the same carbon backbone. Let's go and check it out. So these intermolecular reactions will form two types of cyclic structure, pyranose and furanose. Pyranose, pyranose means six-membered ring structures. Furanose comes when there's only five-membered ring structures. So let's just discuss how these intramolecular reactions happen. So we call it intramolecular hemiacetal formation in aldehydes. Hemiacetal, hemiacetal formation. So hemiacetal means uh, hemiacetal. So let's just go and check it out. So here this carbon, this hydroxyl group attached to this carbon chiral, the chiral carbon, it has its electron is high, right? um, so what it does is that this extra pair of this lone pair of electrons on oxygen, they attach to this carbon right here, this carbon to this carbon right here, and as it attached, let me take a note. No, let me take a pencil. Okay. Okay, anyway, this lone pair of electrons on this carbon attacks on this carbon, and when this lone pair is shared between this carbon, then this carbon gets like five bonds, so this double bond gets broken, and then this oxygen, this electron pair, the double bond, the electrons of the double bond, they get dispersed on oxygen, and oxygen get a negative mark, negative negative charge. So after that, um, then what happens is this hydrogen over here, as this oxygen now got like three bonds, it's unstable. This hydrogen over here is that hydrogen over here, since it got since it's unstable here to be like this hydrogen gets attached to this oxygen, and the final product is formed. So the same things happen here. This oxygen, this electron pairs on oxygen get attaches to this carbon and forms a bond. Then this double bond gets broken up, and then the electrons are dispersed on this oxygen, and then this hydrogen over here is it's unstable. It forms a bond with this oxygen. So it's discussed in details later. So for now, intramolecular hemi. So here it's discussed in detail. So it go through it again. This this chiral carbon is over here, so you can see these carbons are not shown here, just on its numbers. This carbon here is a chiral carbon. This hydroxyl group, oxygen here, attaches on this carbon, attacks on this carbon, and then forms a bond, this carbon with oxygen, of this is it's shown here. This carbon forms a bond with oxygen, and this H will go and attach to this H, this oxygen, right? So after going to attach to this oxygen, the hydroxyl group can be either positioned downside and hydrogen on the upside, or the hydroxyl group can be positioned on the upside. So when the hydroxyl group is positioned downside, remember we call it alpha D-glucose. Okay. When the hydroxyl group is positioned upside, we call it beta D-glucose. So let's take a note. Mm. when hydroxyl group we call it alpha glucose alpha. Let's keep a blank because this molecule name can be changed. Hydroxyl group is positioned upside. It's called beta and then molecule name. Okay, and then we'll go down. Anomeric carbon is a new chiral carbon that is created through cyclic reaction, cyclization reaction. Anomeric carbon is a new chiral carbon that is created through the cyclization reaction. So here, um, um, this is glucose and then formation of cyclic hemiacetal and new chiral carbon. So here, anomeric carbon is a new chiral carbon that is created through cyclization reaction. So what happens here is that this hydroxyl anomeric carbon is a new chiral carbon that is created through cyclization reaction. So when cyclization or formation of cyclic hemiacetal happens, the new carbon created it forms a bond with oxygen and hydrogen and hydroxyl group, and the almost existed mole part, we call it new chiral carbon. So the now this actually this chiral carbon, it was first this chiral carbon of the molecule was this fifth carbon, but now it is a carbon 1. Okay, so anomeric carbon is a new chiral carbon that is created through the cyclization reaction. Okay, cyclic structure formation in fructose. So now let's check what happens in fructose. So instead of the aldehyde, it was a it was a ketose group here. Now there's a carbon here, the chiral carbon. No, it's the chiral. To believe it's this carbon. No, not that carbon, right? So when we take this, let's find out its chiral carbon. So I think that fructose didn't have chiral carbon. No, yeah, fructose don't have a chiral carbon. No. I think chiral carbon is this. No, it's not a chiral carbon. Yeah, this is the chiral carbon of fructose, right? Um, so what happens here is this oxygen this hydroxyl group is activated, and the chiral carbon does not participate in the reaction, but this hydroxyl group within the at the terminal end participates for the reaction, and then no, actually it's not even that thing, right? It's like 5 5 is here, 6 is here, 4 is here. So the bond with 5, this is the chiral carbon here. Is it the chiral carbon here? Currently here. So why is it the chiral carbon here? So when we take this got C Yeah, CH2OH. Yeah, okay, this is a chiral carbon. The fifth one is a chiral carbon because this part is a CH2OH, and yeah, this is the there are like many chiral carbons, but what we take to be here is that the terminal end carbon, the adjacent carbon to the terminal end carbon, we take that. Always remember, it's not the chiral carbon we take. It's always a chiral carbon, but we take the adjacent carbon to the terminal carbon. Okay, adjacent carbon to the terminal carbon, primary alcohol carbon. Adjacent carbon to the terminal primary alcohol carbon, we take it and terminal alcohol carbon, we take it and yeah, that carbon, here this one, participates in this one participates in so here you can see that this fifth carbon is the carbon and adjacent carbon, the terminal primary alcohol carbon. So the oxygen attached to it will the lonely pairs of that oxygen actually this is a B molecule, so this is like some sort of an electrolyte will I don't know, like an element with more electron density will attach on this hydrogen, and that hydrogen, so that in that within that attaching this oxygen will this lonely this bond of bond of electrons in this hydroxyl group will attach to this will attack on this carbon, right? So that carbon will then on this oxygen bond will then um, it will then you know what it will attack on this hydrogen, and then this A will go away. It will attach on a HA. HA is usually it describes an acid, okay, acid, like a uh, poor acid, and then that H plus will be taken to this oxygen, will be attached to this oxygen, right? So this oxygen, this OH will be there, and then this OH should be the air, and then a bond will be formed with this oxygen and carbon, and then this hydrogen will be taken with B, will be taken by B. So HB is eventually formed here, right? Yeah, so that's what happens here. So you have to actually put this AB acid into this medium in order to happen this, in order for this to take place. So then this is a reversible reaction, and then it will form these two types of these two types of molecules, in which this hydroxyl group, the newly formed hydroxyl group, I mean this oxygen, this oxygen over here forms this hydroxyl group, and this in this is the first one, the carbonyl oxygen, right? Carbonyl oxygen in this carbonyl in this ketose group, that when this hydroxyl group is upward, it's called beta fructofuranones. When it's downwards, we call it alpha fructofuranones. Okay, so usually 23% of this fructofurans is formed, and then 5% of this alpha fructofurans is formed in the medium. So let's go back to the glucose one and check the percentages. You can see that always the beta one is formed in a large amount, right? So that's about it. Now let's talk about anomers. Anomers is actually those alpha D glucopyranones and beta glucopyranones. Those two types of molecules formed by the reaction due to the positioning of the hydroxyl group. Okay, let's just take the definition. Intramolecular reactions between the carbonyl carbon. Intramolecular reactions between the carbonyl carbon. Uh, Intermolecular reactions between the carbonyl carbon and hydroxyl group permits two types of anomers. They are the alpha type and beta type. They differ only at the anomeric carbon. intramolecular reaction between the carbonyl carbon and the hydroxyl group permit two types of anomers, intermolecular reactions between the carbonyl carbon and the hydroxyl group. So actually the carbonyl carbon is the carbon that's in the ketones or the aldehyde bond, the double bonded with oxygen. That is called a carbonyl carbon. Okay, so carbonyl carbon is the carbon in the, okay, let's say carbonyl carbon, and the hydroxyl group permits two types of anomers. They are the alpha type and beta type. They differ only at the anomeric carbon. So the difference is at the anomeric carbon. So anomeric carbon is the carbon of the carbonyl group. Okay. After the cyclic reaction, the carbon of the carbonyl group is called the anomeric carbon. Let's just take a note of it. After the cyclic reaction, the carbon of the carbonyl group now is known as the anomeric group. After the cyclic reaction, the carbon of the carbon is known as the anomeric carbon. So anomeric carbon is anomeric carbon now. Due to this anomeric carbon, this anomers, they differ at only the anomeric carbon, right? The alpha type and beta type, they differ at only the anomeric carbon. It's also right that anomeric anomers only differ at anomeric carbon. Okay, when the hydroxyl group on the anomeric carbon of a ring structure of a D sugar is below the plane alpha anomer, when the hydroxyl group on the anomeric carbon of a ring structure of a D sugar is below the plane alpha anomer, when the hydroxyl on the anomeric carbon of D sugar is above the plane beta anomer. When the hydroxyl group on the anomeric carbon of a ring structure of a D sugar is below the plane, it's called the alpha anomer. So, hydroxyl on the anomeric carbon of a ring structure of a D sugar is below the plane, it's called the alpha anomer. So when the hydroxyl on the anomeric carbon of a ring structure of a D sugar is below the plane alpha anomer, when the hydroxyl on the anomeric carbon of a D sugar is above the plane, it's called a beta anomer. Right, when the hydroxyl on the anomeric carbon of a ring structure of a D sugar, when the hydroxyl on the anomeric carbon of a ring structure of a D sugar is below the plane, it's called the alpha anomer. Okay, so when the hydroxyl on the anomeric carbon when the hydroxyl on the anomeric carbon of a hydrogen structure of a D sugar is below the plane, it's called the anomer. When the hydroxyl on the anomeric carbon of a D sugar is above the plane, it's called the beta anomer. So this is how the proper definitions are given. You have to say that it's when the hydroxyl on the anomeric carbon of a D sugar is above the plane, it's called the beta anomer. So it's like you have to tell it relative to the plane, okay? Uh, that's how we properly defined. Okay, let's move to the next slide. The beta anomer of a D-ribose. So this is beta anomer of a D-ribose. This is a this is a D-ribose. It's a beta deoxide to beta 2-deoxyribose. So what has happened here is that um, so instead of a hydroxyl group, you can see an H here. Oh, I don't know what it is. It's a slide that should be given a citation. Okay, now it's given the the explanation here. Um, this is beta D glucopyranose. So here, let's go and check this. What does beta anomer of D-ribose mean? Beta D-ribose, beta 2D deoxyribonucleic D-ribose. So beta D-ribose means does not exist free in the cell. Biological importance as phosphate ester enters the structure of nucleosides, nucleotides, RNA, coenzymes, and metabolic phosphate ester enters in the structure of nucleotides, RNA, coenzymes, metabolic intermediates in the pentose phosphate cycle. So what's the biological importance of beta D-ribose is that as phosphate ester enters in the structure of nucleotides, RNA, coenzymes, metabolic intermediates in pentose phosphate cycle. So beta-D-ribose acts as metabolic intermediates in pentose phosphate cycle when phosphate ester enters in the structure of nucleosides, nucleotides, RNA, and coenzymes. So ribose means actually it means 3 carbons, right? So when there are 3 carbons, actually ribose is 5 carbons. Yeah, 5 carbons. Now we are talking about pentose. It's not about hexoses. So when we talk about these pentoses, these ribose, there are 2 types of ribose. That one is beta D ribose, beta 2D deoxyribose. So you know what are these, right? This is RNA. Deoxyribose means DNA, right? This molecule is important in forming RNA, while this beta deoxyribose is important in forming DNA. Okay. So what's important? Beta D ribose? It means RNA simply as phosphate ester enters in the structure of nucleosides uh, in the structure of nucleosides in the RNA coenzyme intermediates in pentose phosphate cycle. So, pentose phosphate cycle, we are really not aware of it yet, but we have to keep in mind that the importance of this, what's it, beta D ribose, beta D ribose is that when phosphate ester uh, enters enters uh, enters in the structures of um, enters the metabolic intermediates in this ribose acts as a metabolic intermediate in the structures of metabolic intermediate in pentose phosphate cycle that will take place in the structures of pentose phosphate structures pentose phosphate cycle uh, in the structures of um, nucleotides, RNA, coenzymes, Okay, so the other thing is beta 2 deoxyribose deribose. In the structure of deoxyribonucleosides and nucleotides, structural monomers of deoxyribonucleic acid. In the structure of deoxyribonucleosides and nucleotides, structural monomers of deoxyribonucleic acid. So, does not exist free in the cell. Biological impulse as phosphate enters, enters in the structure of nucleosides, nucleotides. Okay, so now it's Ba2 deoxyderivase, that means actually DNA. In the structure of deoxyribonucleosides and nucleotides, structural monomers of deoxyribonucleic acid. Yeah, so simply this beta D ribose makes up for RNA and even nucleosides and nucleotides, coenzymes and so now we have beta 2 deoxyribose, beta 2 deoxyribose. What it does is structural monomers of deoxyribonucleic acid. In the structure of deoxyribonucleosides and nucleotides, structural monomers of deoxyribonucleic acid. So honestly, this 2D deoxyribose means what? Structural monomers of deoxyribonucleic acid. Okay, so let's move to the next one. Physical properties of monosaccharide. So what are the physical properties of monosaccharides? Colorless crystalline compounds, readily soluble in water. Their solutions are optically active. So monosaccharide, they are colorless and they are crystalline compounds and they are readily soluble in water and the solutions are optically active. So what's important is that solutions are optically active. It's kind of very new for us, right? Optically active solutions in monosaccharide. It's a physical property of optically active solutions. It's a physical property of monosaccharide. So let's move fast to the next slide because there's like a tons of things to discuss. Glucose. So when we talk about glucose, this is structure of glucose. In our glucose, there's about 6 carbons here, and then the aldehyde group, and then the hydroxyl group attached here. So you can see that. Okay, this is a right. This hydroxyl group attached to the terminal carbon, primary carbon, alcohol carbon, and adjacent carbon to the primary alcohol carbon is on the right side. So it's D glucose, right? The main monosaccharide found in the human body. Glucose synthesized from non-carbohydrate precursors in animals. Glucose is synthesized from non-carbohydrate precursors in animals. Mainly produced by them. So how glucose is synthesized in animals is by non-carbohydrate precursors, mainly produced by the photosynthetic process occurring in plants. In animals, how glucose is produced is by non-carbohydrate precursors in animals. I don't know what it means. So let's see. Starch is storage form in plants. Glycogen storage in animals. Glycotic pathway. So what is called glycotic pathway is that energy in the form of ATP. So we break down glucose and we store this energy in ATP. So glycotic pathway is energy in the form of what? So what this is glycotic pathway does is it turns this energy in glucose to ATP. Breaking down of glucose will be making ATP. So, a major structural component of most of the disaccharides, trisaccharides, and polysaccharides present in all cells and all body fluids except in the urine. So, the only place where you can't find the body fluid, the only body fluid and cells where you can't find glucose is urine related cells and urine. Okay, urine. Uh, By the oxidation in the liver, glucose is converted to gluconuric acid, which play an important role in solubilizing toxic substances in the blood and thereby enhancing its excretion through urine. In solubilizing toxic substances in the blood, important role in solubilizing toxic substances and thereby enhancing its excretion through urine. So, if blood, I mean, the stuff, if these this toxic substances in blood is directly passed to the urine, um, it might cause some problems. But now this glucose converted into glucouronic acid, glucouronic acid, glucuronic acid, curonic acid. In liver, and then this glucuronic acid do is what this glucuronic acid do is play an important role in solubilizing toxic substances. Glucuronic acid play an important role in glucuronic acid play an important role in solubilizing toxic substances. Glucuronic acid play an important role in solubilizing toxic substances. So glucose, gluconic acid play an important role in solubilizing toxic substances in the blood. So it is less harmful when it's excreted to the urine. Okay, less harmful when it's excreted to urine. Okay, fine. Now let's move to the next monosaccharide we have to discuss, and that is galactose. So not found in the free form in nature in large quantities, synthesized by the body, galactose is mainly obtained through the hydrolysis of lactose, found in very minute amounts in blood, CSF, and urine. So CSF is cerebral spherical fluid, and it's a very found very minute amounts in blood and and urine. So not found in free form in nature in large quantities. So this is the main structure of glucose. The difference here is that you can see this hydroxyl group. Two of the hydroxyl groups are on the right side, and also it got an aldehyde, and the aldehyde is on the edge, right? So let's compare this with the glucose molecule and check. Yeah, it's actually a portion of this take the portion of this hydroxyl groups maybe. I'm not sure though. Don't know what's the difference here. uh, what's the difference here? I do believe it's a portion of these hydroxyl groups. Yeah, portion of these hydroxyl groups, because you can see these hydroxyl groups, two of them in the middle, two of them on this side, right? So this is the galactose. Major structural unit found in some disaccharides. So now we're talking about galactose. It's a major structural unit found in some disaccharides, such as lactose, and also complex lipid cerebrosides, and in complex carbohydrates, mucopolyglucides. Okay, so where is galactose found? We can say give some examples. Galactose found in galactose found in one thing is in lactose, it's a disaccharide, and the second thing is complex lipids, so cerebrosides you might get them in MCQs or somewhere. Cerebrosides, it's actually a complex lipid. And then the third one is cerebrosides, and the third one is complex carbohydrate, that means mucopolyglucides. Mucopolyglucides. Okay, mucopolyglucides, that means complex carbohydrates. Okay, the next thing is, in human body, glucose is changed into galactose to enable the mammary gland to secrete lactose. In human body, glucose is changed into galactose to enable the mammary glands to secrete lactose. Component of the antigens present on blood cells. A component of the antigens present on blood cells determine ABO blood group system. Accumulation of galactose. Accumulation of galactose in blood causes galactosemia, led to the formation of cataract. So, components of the antigens present in blood cells. So, actually, galactose is also a component of the antigens present in blood cells. So, you might be asked where you find galactose in the body. So, even in the mammary glands, galactose are present. Why? Mammary glands is another place where you can find galactose. Okay, so I think we should add this to the same note because it might make more sense then. Okay, so then the fourth one is in mammary glands. So mammary glands, how can you find a galactose in mammary glands? Is glucose converted to galactose, and then galactose will be converted to lactose, and this lactose will be secreted from mammary glands as milk. Okay, so this is why why you can find galactose in mammary glands. And the fifth one is that mm, it's found in antigens. Okay, antigens. Antigen. So it's a component of antigen. So you might get this like this. It's found in, whether it's found in blood. Okay, whether it's found in blood. If you are given a MCQ like whether it's found in blood, yeah, you have to say it's true. Component of antigens in blood on blood cells, right? So okay, antigens are found on blood cells or on blood cells. So thereby it helps to determine the AB of blood group. So function of galactose here is determine the AB of blood group. So it's a function of this galactose. Okay, help to determine. Let's say help to because there can be many other substances that which contribute this thing. The sixth one is um, component of accumulation of galactose in the blood. It causes galactosemia, led to the formation of cataract. So now I'll take another note because it's quite different from the things we discussed before. It's a galactosemia means accumulation of galactose in blood. Galactosemia. Okay, galactosemia, and it leads to formation of cataract. Okay, cataract. And then let's move to the next one. So this is a galactosemia. Sugar with galactosemia. If a galactosemia infant is given milk, unmetabolized milk sugars build up and damage the liver. So eyes, kidneys, and brains will be affected. So unmetabolized milk sugars build up and damage the liver. So there will be kidney damage, enlarged liver, jaundice, cataracts, and brain damage. The treatment for galactosemia is a restriction of galactose and lactose for life. So the next one is fructose. So fructose is that is a seed as among all sugars. Honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose. Found in the free form or in a combined form in natural derivatives, furanose form predominance in combined state. Okay, give a break. So now we are going to talk about fructose. So fructose has a double bond with oxygen. The sweetest among all sugars, honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose found in free form or in a combined form in natural derivatives. Furanose form predominates in the combined state. The sweetest among all sugars, so honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose. Okay, so found in the free form or in a combined form in natural derivatives, furanose form predominates in the combined state. So you can say it's the sweetest among all sugars. Honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose. Found in the free form or in a combined form in natural derivatives, furanose form predominates in the combined state. Sweetest among all sugars, so fructose is the sweetest among all sugars. Honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose. Found in the free form or in a combined form in natural derivatives, furanose form predominates in the combined state. So this is the sweetest among all sugars, CH2OH. Um, Honey, fruits, sugar cane, sugar beet are some of the natural sources of fructose. Found in the free form or in a combined form of natural derivatives, furanose form predominates in the combined state. So I think I kept kind of repeating it one together. But what is fructose? Actually, what's the difference in it? You can say that instead of the aldehyde that was before, just the carbon next to this aldehyde carbon on the glucose, it's I mean like the terminal carbon on the other side, it forms a ketone. It's a ketone group. Ketone here over here. It's a sweetest. Okay, sweetest is fructose, honey, fruits, sugar cane, and sugar beet. The natural sources of fructose. Found in some free form or in a combined form in natural derivatives. So they can be either free form or in the combined form in natural derivatives. That means actually fructose is found in free form. That means or in combined form. So that means free form is just as pure fructose. Also they are available in the nature. Free form or in combined from natural derivatives. Okay. Mm, Furanose form predominates in the combined state. So in the combined state, combined from combined form found in free form or in combined form. At the combined form, uh, the predominance found in the combined form are furanose. Predominates found in predominance found in furanose are furanose um, found in combined form are furanose. Found in the free form in the seminal fluid. So found in the free form in the seminal fluid, a major structural component of sucrose, absorbed much slower than glucose, act as an important intermediate in the metabolism of glucose, glycolysis, and pentose phosphate pathway. So this, this fructose is found in the free form in the seminal fluid. It's a major structural component of sucrose, absorbed much slower than glucose, act as an important intermediate in the metabolism of glucose. Found in the free form in the seminal fluid, a major structural component of sucrose, absorbed much slower than glucose, act as an important intermediate in the metabolism of glucose. So fructose is found in free form in seminal fluid. Um, it's a major structural component of sucrose. We know that, and absorbed much slower than glucose. Why is it absorbed much slower than glucose? I don't know why. And the next fact is that act as an important intermediate in the metabolism of glucose. So uh, fructose is also used as a